Hi everybody, this is Amy from Nexlum Bioscience. I'd like to thank all of our attendees for joining us today for our informational webinar. The topic today will be patient-derived organoids, or PDOs, to model therapy response ex vivo. I'm going to have Scott Kribis, our Director of Applications in Europe, introduce our speaker today. But before we get to that, I would like to remind all of our attendees that they will be muted throughout the duration of the webinar. But if you guys have any questions for us, feel free to type them into the questions bar, and we will have time at the end to answer any questions you have. Also, the session today will be recorded, so everyone will receive a link to the recording after the webinar has concluded, and you can watch the webinar again or pass it along to a friend or colleague if you wish to share the content with them. So I'm going to pass it over to Scott Kribis. As I mentioned before, he is our Director of Applications in Europe, and he will be introducing our speaker today.
Okay, thanks, Amy. Thanks everyone for attending our webinar. Somaieh Hedayat is currently studying her PhD. Her studies are based around trying to investigate how the tissue microenvironment can affect the response of metastatic colorectal cancer cells to antigenic therapies. I first met Somaieh prior to her beginning her PhD, which I believe was something like 10 years ago. Somaieh was working as a higher scientific officer in the Drug Discovery Unit at the Institute for Cancer Research and Professor Sue Eccles' group. Her main focus there was target validation of cancer biomarkers. After gaining experience in vivo and ex vivo biology, she decided to adjust her career path slightly, and in 2016 embarked on a PhD in Kinetic Evaluation group at ICR. Her work was instrumental in the recent science publication entitled Patient Derived Organoids, Model Treatment Responses of Metastatic Gastrointestinal Cancers. So it's with great pleasure that I hand you over to hear all about Somaih's wonderful work.
I'm very excited and delighted to present some of my current data on the patient-derived organoids, as I think this is a very exciting model and a study that I would like to share with you today. So patient-derived organoids are turning to be a very hot topic, and PDOs are intermediate model between the pre-existing in vitro cell line models and xenograft. So the PDOs have been used not only in the field of oncology as well as other diseases which have an essential need for the potential precise medicine. Utilizing a patient organoid culture system, high-throughput drug screening, coupled with genomic analysis from patient-derived organoids and tumor samples could offer a unique opportunity to stratify and identify the effective cancer therapeutic for individual patients. So as a long-term aim, this is what we would like to evidence in a future study with a patient-derived organoid. So to be able to use this platform to promote the discovery of the novel therapeutic approaches, we need to recapitulate the metastatic niche and compare the PDOs drug responses that we observe in the laboratory ex vivo with a clinical response observed in a patient. So to do that, we employ different laboratory techniques and a platform to be able to monitor and observe the drug responses as well as the functionality of each PDO from the individual patient. So as my talk goes today, these are my outline, and I would like to introduce you to some of the points which are important in the study of the patient-derived organoid. First, I would like to give you a background about what are the patient-derived organoids, where are patient-derived organoids come from, are these PDO representative of the parental tissues, and can we recapitulate these clinical responses in vivo? As well as we want to see if we can recapitulate clinical and in vivo responses ex vivo. And on top of that, which is a very important part of these studies, are the fact that we want to see, can we monitor tumor growth kinetically in ex vivo system by imaging? And in here, I'm going to take you through all the success that we had with the Celigo S imaging cytometry. And also we want to see, can we use tumor microenvironment to mimic patient stroma interaction? And at the very end, I'm going to give you a summary of some of my findings and the recent optimization, and we will have some time for questioning. So what are patient-derived organoids? Patient-derived organoids are a mini tumor that form from the tissue that exhibit the intrinsic 3D dimension architecture of the organ that retain their cell-to-cell and cell-to-matrix interaction. PDOs are used in research as a bridge between the cell line in vitro setting and the xenograft. And the PDO culture system, the high throughput drug screening, and all the genomic analysis which are represented platform to generate this direct correlation between the cancer genome and treatment outcome. PDO might identify personalized therapeutic alternative for individual patients for whom a standard treatment has been exhausted or they develop resistance. Where our PDO come from? Our PDO were generated from metastatic chemorefractory colon adenocarcinoma, gastric, and liver cancer. So these PDO were established from the PROSPECT trial, where the sequential biopsies were collected before the start of the treatment and when the patient developed the resistance, as well as at the best response where the patient shows some stable disease throughout the drug treatment. Molecular profiling of the PDO with the parental tissue and archived material from the primary cancer was characterized and compared, and the PDOs were used in the high throughput drug screening and represent and looked at the response to anti-cancer agents, which they were functionally matched with their genetic profile. Also, the patient response to treatment in clinics was compared with ex vivo responses in their derivative PDO. And if you're interested, I stated the recent paper for extra reading if you wish to visit the page. So first we wanted to see, are these PDO representative of the parental tissue? For this study, first we received all the biopsies and we had a 70% success rate in establishing the PDOs from the tumor biopsies. And we compared the morphology as well as the mutational landscape. As you can see from the left-hand side, based on the immunohistochemistry staining of this patient based on a H&E staining, you can see there is a very significant morphological similarity between the organoid and the tissue that they were derived from. And if you look at the mutational landscape on the right-hand side from one particular patient and look at the tissue at the BL, which is highlighted in green, and compared with the organoid, the BL with the two different pathogens, we're having a similar mutational landscape. So as PD, A and B, which are the two different biopsies at the two different lesions, and compared with the organoid of the same organoid of the lesion, we see that the mutational landscape has been preserved throughout the organoid pathology. So next we wanted to see, can we recapitulate the clinical responses in vivo and ex vivo? So to do this, we designed a reverse translational study to be able to define the biology and recapitulate the clinical findings. So we had all our material from our clinical response from the PROSPECT trial, and we wanted to see if we use these PDO that they were individually extracted from the core biopsy of liver tissues of each patient, to see if we can recapitulate all that study and look at the sensitivity ex vivo in a standard 3D system, as well as co-culture system, and also look at the sensitivity in vivo to be able to recapitulate the same finding outside the patient's body. So first we started via recapitulation of the clinical response by using a PDO in vivo setting. So in here, I selected one patient who we had the biopsy at the baseline, which they were sensitive to the treatment, and when they progressed and developed acquired resistance. In this model, we inoculate the tumor into the liver of the mice and treat them with the vehicle and VEGFR inhibitor and look at the growth and the survival rate. As you can see from the graph that I presented you here based on the luminescent flux expression, we see there is a significant differences between the BL and PD. And this is based on the number of days that the tumor stayed in the liver. By day 48, acquired resistant organoid growth at the stage at the baseline when it was in 117. So this shows there is a significant differences between the growth. And when we compare the overall survival of these mice, we noticed that there is an increase of the survival rate in 1021 BL, which is the baseline in comparison with the progression. So in here, we show that our data is our in vivo data and clinical data are correlating strongly. And now we wanted to see, can we kinetically evaluate the PDO growth rate and the drug responses ex vivo? And can we use the Celigo S imaging cytometry platform to study PDO functionality? So to do that, first, we had to know what is the best condition for these organoids to grow. So when we received the biopsies, we main digest and mechanically force these biopsies in order to release the cells into the solution. And once the dissociation took place, we collected it, we pellet, and we suspended the organoid into 120 microliter of the growth factor reduced Matrigel, which was 100% Matrigel, which is a perfect environment for the organoid to grow. Plated them on the 24-well flat bottom cell culture plate, which was provided by Corning. And then once the Matrigel was solidified after 20 minutes in incubator, we added the 500 microliter of completed human organoid media. So the complete media was subsequently refreshed every few days. So this method shows that this is the best condition for the PDO in regards to growth expansion and biobanking. And I've just presented you here with some of the organoids, images of the organoids that they've been taken under the same magnification. As we can see, these organoids are very heterogeneous. So different organoids morphology present the 3D structure that are some dense and compact, some cystic and monolayer around the central lumen. So having this material, we wanted to find a platform to be able to look at the functionality studies of these organoids. So we use Celigo S imaging cytometry. Why Celigo? So we had different platform here, but we wanted to look at something that gives us the best in-class bright field imaging capacity, and also Celigo offers 4 channels, which was quite important for us to be able to detect the different fluorescent light. So Celigo has got blue, green, red, and far red. It's very multiplex and highly speedy. It's fully automated imaging system, and it's got a fast quantification reading, which it can be used for the suspension, adherent, tumor spheroid, PSCs, and cancer stem cell clonies. The system image every cells in every well without any well edge effect. And for 96 well plate, it takes less than 5 minutes to image and a lot less than 1 minute to analyze the whole well. So on top of all of that, if you wanted to use for the high throughput screening, we identified that Celigo system is compatible with the automation platform for a full integration and high throughput screening for further studies. So to be able to perform my experiment, I needed to use the method that's already been published to look at the drug screening and the growth rate of the organoids. So the current published method is not very good when it comes to imaging, just because the organoids do grow and they grow the best microenvironment, which is the 100% matrigel. So it's a pure matrix matrigel. And for here, I'm just going to explain to you how does this work. That I think that is not really a good method to study the imaging. So on the day 0, the PDO were harvested, dissociated, and resuspended and counted. 5,000 cells per 30 microliter of the growth factor reduced matrigel, which is 100%, seeded in each well of 96 well cell culture plate. In here, it's used Corning plate. Incubated for 20 minutes in incubator till it's solidified and the media added. After 3 days, media is either being changed for the fresh media or the drug has been added with the right concentration. And this drug has been changed, refreshed every 3 to 4 days, depending on the growth rate of the organoids. And up to here, no imaging can take place. And the endpoint study is a cell titer blue to look at just the viability. And the reason for that is, if you look at the imaging here, there are some complications that the images cannot take place. So one of the biggest complication is a focusing issue, because some of the organoids are presented at a different stack of the matrigel, because the thickness of the matrigel, it doesn't let any system to dissect them completely. And also, if you look at the well, we see that the Corning well is slightly dome shaped. So we get the center, we get the empty centers, and also majority of the matrigel, they move on one side and it makes the other side empty. So for us, we wanted to overcome this issue, to be able to get more information out of this organoid over the period of time and look at the morphology. So we developed a new approach. So the new approach is very similar, with the difference that after we did the harvesting and resuspending the cells, we use the Griner plates, which are high, they have a high clarity, they are a macro clear bottoms, and also the plate is extremely flat bottom. So in here, we use the same volume of 100% growth factor reduced matrigel and did the 4 different cell seeding density. Once the cells they were plated, the plates were centrifuged at 1000 RPM for 1 minute and incubated for 30 minutes in the incubator, and the later media added at the volume of 70 microliter. So on the day 3, replenished with 50 microliter of the fresh medium containing the complete human medium, and the plates were imaged every few days to make sure all the PDOs were captured. And all of that, out of the 96 well plate, took approximately 10 minutes for imaging. And at the end point, I performed a cell titer glow 3D viability assay. So as you can see from image, on the day 3 and day 10, and the different concentration of the organoid seeding, we see that absolutely every organoid has been captured. So for this assay, I used the single cloning verification application, and for the data analysis, I used the confluency. So I'm going to show you here the Celigo measurement based on the percentage of the confluency, where you can see here on our final measurement for each was based on the 100% confluency as well as cell titer glow. So we see on the day 3 and the day 10, there is a very strong correlation between the decrease of the confluency based on the cell seeding density. And if you compare that with the cell viability, cell titer glow, you see the same trend. So we were quite happy with this assay. So we solved the problem that we had in regards to 100% matrigel. So next, I wanted to see, can I recapitulate or can I form the organoids, use the organoids and do the 3D spheroids? And the purpose of the PDO in the 3D spheroid formation is to be able to assay the organoids for the migration assay, invasion, co-culture studies, and a high throughput screening. So for this study, I used the U-bottom plate from the Nexcelom, where the PDOs were harvested and 150 microliter of the 2000 cells per 200 microliter were seeded in the ultra low bottom, attachment 96 well tissue culture plate, which was provided by Nexcelom. Plate incubated and in the incubator for 4 days. And on day 4, 100 microliter of the media was removed and replaced with 100 microliter of the fresh media. Media was refreshed every 3 days, and the plates were imaged by Celigo every 3 days, which this imaging took less than 3 minutes per plate. And the cell titer glow 3D cell viability assay performed at the end of the study. So in here, for me to prove that this works or not, so I used my PDO from the study that I'll show you earlier on from in vivo study. So I use the same PDO of the same patient from the baseline and the progression when they acquired resistance. I just seeded them in the U-bottom, spin the plate, image them with a Celigo, with a Celigo imaging system. In here, we compared the tumor growth in 3D, and we identified that our baseline PDO grows significantly slower than the progression, and this data mimic what we observe in an in vivo setting. So then again, I take another box, and we wanted to know, what if we want to use these organoids to grow them as a single organoid for CRISPR purposes, as well as other studies such as looking at the heterogeneity for sequencing and the microarray studies. So I wanted to know if I can use the organoid with the different concentration in U-bottom plate. So in here, I started my concentration of the organoids at 200 all the way down to 1 organoid and monitored them over the time. And as you can see from the top one, on the day 8, I do have the organoid with the different sizes, and when it gets to 1, it's very small. So I zoomed in, and I've got presented you here at the different timing to see what happened to the organoid over the time. As you can see, by 16 hours, they start dividing, and they come together, 52 hours, they divide again, they get separated, and by day 8, they come together and form a really strong organoid. So again, in this part, I was quite happy, but again, I have to say, I have to add something here. Not every organoid is capable of forming a single spheroid. So there are some that is impossible, some are growing faster than another. It's quite heterogeneous throughout the whole organoid. Next, I wanted to look at the migration study, because this is another point that we identified in our clinical setting, that our organoid from progression study is a lot more migratory than the baseline. So I wanted to set up the right matrix and the right assay to monitor their migration. And as you can see here, from day 0, I see that I coated the plate with my appropriate percentage of the matrix, added the organoid on top, at the media, and monitored them for 11 days. And what I identified is that the PDO are also capable of migrating within a matrix when seated on top with appropriate concentration, as well as the progression PDO are highly migratory in comparison to the baseline, which again, this was something we observed in a clinical and in vivo setting. So as a part 1 summary, I have developed a method where PDOs can be imaged kinetically at their optimum matrix condition. Also, the images are acquired by using a single clone formation application and analyzed by confluence parameter. By incorporating hypoxia and nutrition deprivation, cell-to-cell, and the cell-to-matrix interaction, I have developed a PDO 3D spheroid, where the image of the PDO 3D spheroids were achieved by using a tumor sphere application and average diameter or the volume calculation to compare the size and the volume of the spheroids at a different time interval. The single organoid formation was successfully completed for cloning purposes and heterogeneity studies, and also using an appropriate percentage of the matrigel, or possibly I should add here, by using a right matrix for depending on where the organoid come from, we can recapitulate the migration capability of organoid in an ex vivo setting. So for the second part, I wanted to see, can we mimic tumor-stroma interaction ex vivo? So this is something that it was quite important to recapitulate the same tumor microenvironment outside the patient body, because we knew that these organoids came from the liver, and the liver doesn't contain just the tumor cells, so they contain stroma and the tumor vasculature. So by getting some information from our pathologists, we got some scoring from our responder versus non-responder patients. So from this data, I wanted to see, by incorporating a microenvironment into the ex vivo co-culture, this might help me to recapitulate that complexity of the solid tumor that we have in patients. So I selected the right percentage of the endothelial cells and the fibroblasts. And what I did, I used for this study, I used the organoids again from the same patient at the baseline and at the progression. I performed the staining with the DID, which is a cell tracker, and for HEWEC and cancer-associated fibroblasts, I used the more stable tagging, which in here for the HEWEC, I use a GFP, and for cancer-associated fibroblasts, I used the RFP tag, which they were stably tagged here. So from this production of these cell panels, I set up my PDO 3DS for a co-culture system where I used 10% HEWEC, which they were GFP positive, 30% CAF, which they were RFP positive, and 60% of my PDO, which they were cell-tracked with DID. So as you can see here from the left-hand side, when I put them all together after 4 days, I get the, based on fluorescent imaging, I get the really strongly and tight spheroid. And if you look at the confocal microscopic imaging, you can see how they are all incorporated together in the spheroid. So the aim of this was for me to understand of the mechanism of resistance, and also to be able to identify a new biomarker to use in a combination therapy based on the co-culture microenvironment. So what I did for this, to just get some validation out of the way, for here I used all of my cells that I tagged already, pull them together, use the appropriate concentration, and I monitored the spheroids over time, over 10 days. So I saw the significant increase of the tumor growth in the progression. As you can see here in red, they grow a lot faster than the baseline. And when I did the when I compared with my previous data, as you can see, the data is correlating very strongly with my in vivo studies. But this wasn't it. So I wanted to develop a model that I can use for the drug treatment. So I wanted to develop this 3D spheroid co-culture system and to be able to individually identify the cell population that they get hit by the treatment. So I use bevacizumab as a treatment in here for my validation study at the different concentrations. So as you can see here, I'm just presented you with all the fluorescence. So I did far red for my patient-derived organoid, for endothelial green tagging, and for CAF is a RFD. So PDO 3D were seeded on the 96-well Nexcelom plate, and the plates were imaged on the day 0 and the 4 days after the treatment. And the whole 96-well plate took about 12 minutes, and I used a tumor sphere 1, 2, 3, and mask application. So from here, I show you after doing the imaging based on the average diameter, just looking at the spheroids formation, you can see I don't see significant differences in the reduction of the spheroids. But if I look at the average fluorescent integrated intensity of every individual cells that I added into my core culture, you can see that there is a very strong trend of the reduction after the treatment at the highest concentration of the drug after 4 days. So the average diameter doesn't provide much information about the effect of the compound, where the average fluorescent integrated intensity gives me that information that I'm lacking. So by performing this assay, you can do 4 channel of which is mask, fluorescent, far red, green, and red, and all the analysis in less than 15 minutes with Celigo imaging system. So as a part of summary, I'm going to finish my talk today. It's based on immunohistochemistry scoring to identify the appropriate co-culture population to recapitulate our patient mini tumor environment by incorporating a 10% endothelial cells, 30% cancer-associated fibroblasts, which they were derived from colorectal cancer, and 60% of the patient-derived organoids. The patient-derived organoid 3D spheroid co-culture system form a 3D architecture of the tumor, which mimic the tumor microenvironment, and they were successfully transduced for each cell population. Celigo S 5 channels was able to acquire the fluorescent intensity of 3 of the channels that I was interested in on one single application in less than 15 minutes. And my recapitulation, clinical and in vivo responses in ex vivo was all shown based on the same patient at the progression, which had a it had a significantly higher growth rate when it was compared with the baseline, which they were sensitive to the treatment. And the fluorescent intensity also allowed to identify the cell population, which are able to be inhibited by targeted therapy rather than just analyzing the whole spheroids. So at the end, I would like to thank my team. I'm part of the molecular pathology team at Institute of Cancer Research under supervision of Dr. Nicola Valeri. And a huge thank you to George, who developed all these organoids over a few years, and all the clinical team from Royal Marsden. And also a huge thank you to Scott for all his support throughout nearly 10 years working together. And I am open to any question. Thank you.
Thank you, Somi. I think that was a wonderful presentation.
Thank you.
So as she mentioned, this is time for questions and answers. So all of our attendees, just a reminder, you are muted. But if you do have any questions, type them into the questions bar, and I will read them out to Somi and Scott so they can answer the questions. I will give everyone a minute or so to type those in. Okay, so we do have one question that has come through. The first one states, which methods do you think is better for drug screening, organoids with matrigel or spheroids?
If you're asking my personal view, if you just want to do organoid alone, not with a co-culture, I would stick to what is the best condition for the organoid growth, because that's what we want. We want them to be in their best health stage to be able to do the drug treatment, which is 100% matrigel. And now we have this assay developed where you can kinetically monitor them over the time, as well as doing an endpoint, which it would be much more appropriate for looking at the effect of the drug rather than steroids. And another reason for steroids, I'm trying to avoid just a single PDO steroid, is that some of the organoids grow really fast. And when they go into the 3G spheroids format, the center part slowly become hypoxic, and avoiding that, it can be quite complicated. Where with the flat bottom, you can increase the days of your treatment up to 12, 14 days, where with the spheroids, at some point, you really have to stop it because of the necrosis and cell death within the spheroids.
Great, thanks. Another question has come through. It states, do you usually normalize average fluorescence integrated intensity with the size of the PDO?
I mean, no, I don't. I don't go with the size of the PDO because I have control there as well, which are not treated. I tend to go with a control, but because size of the PDO, as I show you, is not really relevant to actual spheroids, because there's a 60% of population of the organoid, 30% calf, and 10% HUVEC. So sometimes I go, like I do a few different ones actually. So I do either based on day 0 or based on the control. So there are a few ways you can do this.
Great, thanks. Next question. Have you thought about using any other imaging technology for your assay?
Yes, actually, yes. I spent a lot of time to get this correctly. So I use other system, and as you know, I'm at ICR, we do have so many different platforms that would be quite useful for 2D studies in a specific. But when it comes to spheroids, just because of the speed and the plate type, Celigo offers the best, because the images are absolutely perfect. And if I want to move on to high throughput screening, I would highly recommend Celigo. Just again, as I mentioned before, it's the speed and it's the fact that it can be attached to the robot, and you can do it in a high throughput level. But specifically for my study, I needed a platform that's got more than just 2 fluorescent lights. So I was looking for 4 channels. And Celigo was the one, so as confocal. But if I wanted to do a screening for more than 2 plates, confocal, I just have to sit in with a confocal microscopy for at least 14 hours per day to be able to get all the images. Where Celigo for 2 plates can do the whole thing under 15 minutes for me. So that's why personally, I'm more drawn to that, just because once I start my therapy and once I start my combination, it's going to be more feasible for me.
Great, thank you. Next question. Why do organoids form in matrigel and not in the ultra-low attachment U-bottom plates?
That goes back again to their tumor microenvironment, really, because as you remember, all the publication that is being published out there, they show the organoid grow best in 100% matrigel. So imagine if you take that away from them, they may not come together. So they lose their cell-to-cell contact and they lose their cell-to-matrix interaction. that's why majority of the organoids that we work with, they grow the best with matrigel. But again, in my case, I replaced the matrigel with the cancer-associated fibroblasts. I replaced it with a little bit of endothelial cells. That brought them together to give a right microenvironment. So, I mean, going back to the presentation again, matrigel is the best if you want to have the organoid just by themselves, just because it brings them together, gives them more realistic tumor microenvironment condition. And if you want to go for co-culture, obviously, you know, other stroma, tumor stroma interaction would help as well.
Great. Next question. Have you ever thought about controlling oxygen in vitro?
Do you know what? With the organoids, no, I haven't. So no, not yet, I would say, put it this way, just because that part of the study hasn't been part of my project. But yeah, I mean, it's a very valid point, but because my project is focusing at the moment on angiogenesis and migration and invasions, so I haven't moved on towards that yet, but definitely in future, that will be something to look at the hypoxia and also different ingredients of the oxygen, which can make a huge difference in migration capability as well as the growth of the spheroids.
Okay, I'm not seeing any other questions come through, so I suppose that we'll conclude our webinar for today. If any of our attendees think of any other questions that they didn't type in for us now, oh, we actually just got one more question that came through. Sorry, everybody, we did just get one more. So I'm going to read it out. It says,
sure.
Did you test other matrices such as collagen for your migration assay?
Okay, so, you know, as you notice from slide number 20, I didn't state it with what matrix I used. So this matrix I used, it wasn't matrigel. So I used the human collagen because I wanted to recapitulate the same environment as the patient, because these organoids were derived from the liver. So yes, I used collagen 1, 2, and 4, because these are the ones that are highly expressive in the liver tissues.
Thank you. I don't see any other questions coming through, but we did have a comment that has come through from one of our attendees wanting to congratulate our speaker today, saying that the work you do is really amazing. Just wanted to pass that along to you, Somaieh.
Thank you very much. I appreciate it.
So I will give everyone a few more seconds to see if we get any more questions that come through. So just hang tight for just a minute.
Sure.
All right, thank you, everybody. I'm not seeing any other questions that have come through, so that will conclude our webinar for today. If any of our attendees think of any more questions later on, feel free to reach out to us at support@nextlim.com, and we'd be happy to pass those questions along to Somaieh and Scott to answer those for you. As I mentioned earlier, the webinar today was recorded, so everyone will receive a link to that recording after the webinar has concluded, and you can watch the webinar over again or even pass the content along to a friend or colleague if you wish to do so. Thank you, Somaieh and Scott, for joining us today and for presenting your wonderful experiment and assay, and thank you for all of our attendees for joining us, and I hope everyone has a wonderful day.